Students, now we are going to discuss about your folic acid, which is also your vitamin B9. If you see the structure of folic acid, it consists of pteroic acid group and glutamic acid. Again, in pteroic acid, you have pteridine and para-amino benzoic acid. Regarding metabolic role of folic acid, the active form is tetrahydrofolic acid. How are we getting tetrahydrofolic acid from folic acid? It is with the help of this enzyme dihydrofolate reductase. Folic acid first gets converted into dihydrofolic acid with the help of this enzyme dihydrofolate reductase, and again dihydrofolic acid getting converted into tetrahydrofolic acid with the help of this enzyme dihydrofolate reductase. The reduction will happen at fifth and eighth position of nitrogen atom. The main role of folic acid is with the help of one carbon groups. What are these one carbon group, students? They are methyl group, methyl, methylene group, formimino group, where you will see this thing in the conversion of histidine to glutamate, and hydroxymethyl group, methinyl group, and formyl group. Now we will see different sources from which these one carbon groups are being produced. In the metabolism of serine, which is getting converted into glycine, tetrahydrofolate is getting converted into N5 and 10 methylene tetrahydrofolate. And during the glycine cleavage, tetrahydrofolate is getting converted into N5 and 10 methylene tetrahydrofolate. During histidine getting converted into glutamate, here THF is getting converted into N5 formimino THF, where this formimino group is being taken by tetrahydrofolate, so that N5 formimino THF is formed. And during tryptophan metabolism, in tryptophan metabolism we have three pathways. One is kynurenine pathway, one is serotonin pathway, one is indole pathway. Particularly during kynurenine pathway, THF is getting converted to N5 formyl THF. All these things are interconvertible except methyl THF. Now we will see the utilization of these one carbon groups in different things. One is during the synthesis of methionine from homocysteine, where there is a transfer of one methyl group from methylcobalamin, so that methionine is getting produced. Now for this methyl group transfer to B12, it is being helped by methyl THF. So if there is any deficiency of B12, what is happening is this folic acid is getting trapped in the form of methyl THF, because students, as we told, methyl THF, it cannot be converted into any other form. Got it, students? So vitamin B12 deficiency will lead to folate trap. Now during the synthesis of purines, IMP is being formed from N5 and 10 methylene THF and N5 formyl THF. So from IMP we are getting AMP and GMP. From AMP we are getting ATP, and from GMP we are getting GTP, which are required during DNA and RNA synthesis. Now during pyrimidine synthesis, DUMP is getting converted into DTMP, where N5 and 10 methylene THF is getting converted into DHF, and again it is getting replenished by the enzyme DHFR, dihydrofolate reductase. For the formation of formyl methionine tRNA, this formyl THF is required. This thing is helping for the protein synthesis in prokaryotes and in mitochondria. During Serine synthesis from glycine N5N10 methylene THF is being used. If we see the deficiency manifestation, students, it will cause megaloblastic anemia. Why megaloblastic anemia, students? It is mainly because since we can able to see this thing because of folate deficiency, these things are not produced, so that DNA and RNA synthesis is hampered, and because of that there will be immature RBCs in the blood. They are more size compared to the mature RBCs. And second deficiency is homocystinuria. As we had seen, homocysteine getting converted into methionine, it requires methyl B12, which is indirectly coming from methyl THF. So any deficiency of THF will lead to accumulation of homocysteine, which will be excreted in the form of homocystinuria. Now, birth defects. It will cause, any deficiency will cause spina bifida. Now, how will you assess deficiency, students? Blood levels of normal blood levels of folic acid is 2.7 to 17.0 nanogram per ml or 6.12 to 38.52 nanomole per liter. You can directly measure in the blood, or else you have FIGLU. What is FIGLU? Formiminoglutamate. Histidine, during its metabolism to glutamate, it forms an intermediate compound, FIGLU, formiminoglutamate. This formimino group will be taken up by THF, so that glutamate is formed. Now, because of this deficiency of THF or folic acid, FIGLU will get accumulated and it will be excreted in the urine. Okay, students. Now, we will see the daily requirement, which is 150 to 200 microgram per day, and during pregnancy it is 400 microgram per day. And the sources are yeast, green vegetables, pulses, oil seeds, eggs, etc. Milk is the poor source for folic acid, students. Now, there are few drugs which will act as folate antagonists. What are these drugs? They are sulfonamides, trimethoprim, Septran and cotrimoxazole, methotrexate, and pyrimethrin. Pyrimethrin is used as anti-malarial drug. So this is all about your folic acid, students. B9. Thank you.